My name is Brian McWilliams, and I am the Senior Manager of Product Validation for Bethel Laboratories and Fortis Life Sciences. And today we're going to be detailing some of the work that we did to release a monoclonal antibody against UBQLN4 with this poster here. So UBQLN4 is a protein that has historically been associated with protein turnover in the cell. And recently it has been discovered that through an interaction with a second protein called MRE11, that UBQLN4 is actually having an effect on double-stranded break repair as well. We have had long-term a polyclonal antibody against UBQLN4, and there are others in the market available. However, none of these antibodies are very effective in immunohistochemistry, and so we used our R&D pipeline to develop a new monoclonal to help bridge that gap. Now, for us to come out on the back end of that pipeline and know for sure that that antibody is binding what we say it's going to bind, we are implementing what we refer to as a new pillar-based validation system. This was adapted from a publication in 2011 by a group headed up by Dr. Ullin, discussing some of these sort of techniques or requirements for an antibody to know for sure that it is binding what we say it's going to bind and binding to that protein specifically. So we have those 6 antibody pillars listed out here. We'll talk through them here briefly. The first of these is what we call the independent antibodies pillar, and this basically means that we will be using 2 antibodies that are raised against 2 distinct epitopes of the same target protein, and that those 2 antibodies should behave in a very similar way. We also use what we call a complementary assay, which basically means that 2 assays that are antibody dependent will be able to generate results that are complementary to each other. We also have what we call an orthogonal characteristic, which means that 2 antibody independent assays, such as a mass spec, is producing data that is consistent with the data that we're seeing off of antibody dependent assays. We also have here what we call a biological characteristic, which basically just means that a assay is able to take advantage of the specific biology of one of the target proteins, such as a phosphospecific protein, and that the biology that we are expecting to have happen is consistent with that assay result. We can also use overexpressed proteins or epitope tagged proteins to help show specificity, and we're also able to use what we call a genetic strategy, which is basically a knockout of that gene from the cell line and showing the expected on-off effect of that knockout. So when we went into this project, we started off with 5 unique antigens that were injected and then used ultimately for B-cell selection. We used a full-length protein and then 4 unique peptides scattered throughout the UBQLN4 protein throughout different regions of that protein. And so from each of these antigens, we were able to isolate 46, 9, 8, 7, and 10 total clones, coming out to about 70 total. And then each of those 70 went through a ELISA screen where each of those clones were looked at against the immunizing peptide. Now, one of these clones, this one here that I have highlighted as clone 3, was ultimately the one that we selected to become A700-145. And the rest of the data here is basically showing how we arrived ultimately at that conclusion. So going back to our pillar conversation from earlier, one of our very early stage experiments is comparing with these 2 positive strips here, antibodies that are known to bind to UBQLN4, and each of these strips here represents a Western blot looking at a B-cell supernatant isolated from a very specific B-cell. And so these are basically just very small Western blots showing that each clone is binding roughly to, or not in some cases, roughly to where we are expecting that band to be migrating on a SDS page gel. And again, I've got our ultimate clone here highlighted. You can see a very strong, thick band, comparable or thicker than even the bands that we are showing here as positive controls. Now, once we got through this step of our screen, we're down to about 8 to 10 clones, down from roughly 70. And then one of these 2 positive controls here, we use on this side in a Western blot and a ICC experiment in OVCAR4 cells. And then our clone of interest is shown here, and you can see that we have a very similar and comparable result on Western blot here for both clone 3 and our positive control. And the OVCAR4 staining is very similar for both our new clone and the positive control of interest. So that checks off the antibody or the independent antibody pillar. And now we'll move down into a couple of new ones. This is what we refer to at Bethel as a reciprocal IP, and this will check not only the independent antibody pillar but the complementary assays pillar as well. We basically have 3 different control, or I guess 2 control antibodies and our clone that are all being used in an IP reaction and then run on a SDS page gel and then blotted with our new clone 3 or A700-145. And you can see here that in all 3 cases, all 3 of these antibodies are pulling down the same protein that is being blotted by A700-145. So we know for sure that this band here is in fact UBQLN4, and this checks off the complementary assays pillar. And then as we move down here to the genetic strategies pillar, we have a wild type cell line and a knockout cell line that was provided by one of our collaborators. And you can see again very plainly here that we have a very strong band in the wild type lane and a very blank lane in the knockout cell line. So this again gives us confidence that what ultimately became A700-145 is on target and binding to UBQLN4 very, very specifically. So once we have used those pillars to establish specificity and that this clone is what we say it is, we moved into what we would call some more advanced applications just to kind of see what this could do in a more broad set of experiments. So we took A700-145 and applied it to OVCAR4 cells and looked at a wild type versus knockout flow cytometry experiment. And you can see our clone here compared to a competitor clone here. And you can see that the wild type has 2 very nicely spaced out and distinct peaks here that shift almost completely together once we knock out that UBQLN4 gene. And then compared to here with our competitor clone, we have 2 very nicely distinct and well spaced out peaks that don't quite come together as well, which suggests some degree of non-specificity within that particular product. So here in particular, we were able to show that our clone is in fact superior to the one that was being used previously. And then finally here, we applied our UBQLN4 antibody to a 6-plex multiplex using OPAL reagents. And we're able to show here in orange the presence of our UBQLN4 antibody binding well within this breast carcinoma, and it is co-localizing well with MRE11, which, as you'll recall, is the protein that it binds to help mediate double-stranded break repair, and is also localizing well with gamma H2AX, which is known to be a double-stranded DNA break repair protein as well. So taken at large, we are now able to conclude that UBQLN4 is in fact localizing as expected, and our antibody is performing well, it's performing specifically, and it is performing in a way that is broadly applicable across multiple types of applications. So with that, I am finished, and thank you for your time.